Believe it or not, inside each and every one of us is a hidden chemical universe, a kind of biological dark matter, you could say, and it's filled with molecules that could hold the key to all sorts of future medicines. Now, for decades, this entire world has been pretty much invisible to us, but now, thanks to artificial intelligence, we're finally starting to turn on the lights.
It's a pretty wild question when you stop and think about it, right? I mean, we already rely on products from nature for about 2/3 of our approved drugs, everything from antibiotics to cancer treatments. But what if the really powerful ones, the game changers, are still hiding out there, locked away inside our own biology, just waiting for us to find them?
And that brings us to this amazing term that scientists have borrowed from cosmology, the dark matter of chemistry. It perfectly describes these hundreds of thousands of unknown molecules that are doing something inside us. We can see the effects, we know they're there, but we have no clue what they look like, and we definitely don't know what they do. It's this huge, unexplored territory.
Okay, so to get a handle on this dark matter, we first need to talk about metabolites. These are the tiny molecules that your body makes from, well, everything. The food you eat, the air you breathe, they are absolutely vital. They provide energy, they build cells, they send signals. The best way to think of them is like a live feed, a real-time snapshot of what's happening in your body right now.
And this quote from Ami fellow Russ Greiner just nails the difference. See, your genes, they're like the permanent blueprint for a house. They're fixed. But your metabolites, they're the real-time activity inside that house. The lights turning on and off, the water running. And for figuring out what's going on with your health, that live feed is often way more important.
So where do these molecules actually come from? Well, they're created by something called biosynthetic gene clusters, or BGCs. And honestly, the best way to think of these is as genetic recipes. They are specific chunks of your DNA that have all the step-by-step instructions for building a complex molecule.
And this is where the scale of the challenge just becomes mind-boggling. Thanks to new technology, we've identified millions of these genetic recipes in nature, but get this. We've only been able to connect about 3,000 of them to the actual molecule they create. That leaves a massive, untapped reservoir of potential new medicines that we know nothing about.
So you see the problem, right? We have millions of recipes, but we have almost no idea what they cook. For the longest time, trying to figure this out has been like looking for a needle in a universe full of haystacks.
You know, the old way of doing this was just brutal. It was painfully slow, completely manual, and involved a ton of expensive trial and error that just couldn't scale. But AI completely flips that on its head. It makes the search fast and automated, it can predict which recipes are worth looking at, and it can handle massive amounts of data in a way no human ever could. But here's the thing, you can't just sprinkle some AI on a problem and expect magic. A recent MIT study served up a pretty serious reality check. Nearly 95% of enterprise AI projects failed to actually deliver a real impact. And the reason why? The AI tools were often just one-off experiments, totally disconnected from how the scientists actually work.
This led to a huge realization. The groups that are actually succeeding are the ones that stopped treating AI like a science fair project and started building it into their core infrastructure. It has to be woven into the very fabric of the research process, from start to finish.
So let's see what this looks like in the real world. Our first example is a tool designed to solve that first big problem. We have millions of genetic recipes, but what in the world do they make? For this, meet a tool called BGC-Mac. And the analogy here is just perfect. Think of it as Shazam, but for genetics. You know how Shazam listens to a song and tells you what it is? Well, BGC-Mac reads a genetic recipe, a BGC, and predicts what kind of molecule it's designed to create. And the process itself is surprisingly simple. First, you just feed the genetic code of the BGC into the model. The AI then uses a clever attention mechanism to zero in on the most important parts of that recipe. And finally, it spits out a prediction, telling scientists, hey, this recipe looks like it's for a polykeptide, or you should probably be looking for a terpene. And the results? Well, they really speak for themselves. When you compare BGC-Mac to older, established tools, it consistently comes out on top. That score, an AUROC of over 0.96, is exceptionally good. It means the model is incredibly accurate at classifying these recipes correctly. But honestly, this might be the coolest part. BG-Mac isn't a black box. It's what we call explainable AI. It literally shows the scientists which parts of the genetic code it paid the most attention to when it made its prediction. And for a scientist, that's not just a prediction, it's a brand new insight into how biology works.
Okay, so BGC-Mac is fantastic for classifying the recipes we've already found. But what about all those unknown molecules? How can AI help us find something we've never even seen before?
Well, for that, we turn to another tool called DeepMet. And if BGK-Mac is like Shazam, then DeepMet is basically ChatGPT for molecules. Instead of learning human language from books and the internet, it learned the language of chemistry by studying the structures of thousands of known molecules.
You might be wondering, how can an AI learn chemistry? Well, it turns out you can actually write molecules as text. There's a special notation system called SMILES, which is just a way to represent a complex, 3D chemical structure as a simple string of text.
Let's take a super simple example. The letter C just stands for a carbon atom. If you build on that, a simple string like CC equals OO perfectly describes the structure of acetic acid. Yep, the main ingredient in vinegar. It's the simple language that allows the AI to read and write chemistry.
So the researchers fed thousands and thousands of these SMILES strings into DeepMet. Over time, it learned the chemical grammar, the rules for how atoms are allowed to connect. And once it knew the rules, it could start writing its own new chemical sentences, generating structures for molecules that were totally plausible but had never been seen before.
And the result? It's just staggering. By having the AI generate these new ideas and then checking for them in real biological samples, the research team discovered and confirmed the existence of 36 previously unknown mammalian metabolites. 36! To put that in perspective, finding just one of those using the old methods could take a single researcher months, if not years.
And you can start to see how this all connects, right? This isn't just some cool academic project. These breakthroughs in decoding our own biology have really profound implications for the future of our health and medicine itself.
We're looking at a future with truly accelerated drug discovery, where finding new candidates could take months instead of decades. We're going to get a much deeper understanding of the why behind diseases. This really opens the door to personalized medicine, treatments tailored to your body's unique chemical profile. And of course, by focusing our efforts on only the most promising candidates, we can dramatically lower the cost of research.
When you step back and look at the big picture, what's really happening here is that for the very first time, we are learning to read the hidden chemical language written inside our own DNA. It's like we just stumbled upon a vast, forgotten library of medical texts, and we've only just opened the first book. So the only question left is, what incredible cures are in there, just waiting for us to find them?